Purpose of this video is to finish up the lecture notes on fatigue, primarily finish up peripheral fatigue. Before I do that, I want to do a very quick mini review to kind of summarize what we talked about in class. Again, we talked about fatigue as being caused by two primary factors, both central, which is fatigue related to the central nervous system, and peripheral, which is fatigue in general related to the neuromuscular junction, which includes both the action potential coming from the nerve, the interaction between that and skeletal muscle. Again, as a reminder, central fatigue is fatigue that is possible without the muscles themselves being fatigued. It is in general what we would consider to be labeled as psychological fatigue. So again, this understanding is minimal. We do know that it occurs. Understanding exactly how and why, such as sensory inputs, motivation, pain, etc., can impact psychological fatigue is not super well known, but we do know it occurs. We also talked about peripheral fatigue and the idea that it could come from one of kind of two big sources or hypotheses, if you would. So those two hypotheses are the exhaustion/depletion hypothesis and the accumulation type of hypothesis. So the exhaustion hypothesis gives us this concept that we are exhausting resources, if you would, to kind of make it relatively simple. In general, when we're thinking about those, we're talking about energy-related substrates to produce ATP. So if we have phosphagens or phosphate groups present in low quantities, that can lead to muscle fatigue or peripheral fatigue. The second is the accumulation hypothesis, and this tells us that metabolic byproducts or things that are being produced during exercise that aren't necessarily meant to, but just products that occur at the same time can also cause fatigue by increasing in concentration, therefore doing what we call accumulation. Again, as one might imagine, it's likely a combination of both factors that are going to contribute to peripheral fatigue. And I'll note oftentimes these two scenarios are really hard to tease out, which is more important, because they often occur at the same time. So for example, if we are going to look at phosphocreatine, we can deplete that with really high intensity exercise, but when you do that, you also get high increases in hydrogen ions and a change in pH. And so it's hard to tell if it's creating phosphate or the change in pH that's occurring. That same scenario, same with phosphocreatine, same with ROS. And so many of these things change at the same time. We can look at kind of glycogen, so that gets depleted over really long exercise, and ROS goes up as the duration of exercise occurs. So it's hard to tell which of these ultimately plays a primary role in fatigue, and that's why in this slide you see here we call these hypotheses. It's just the concept. We can't prove either or is the primary driver of peripheral fatigue. So we talked about this. I'll just move through quickly, but looking at the Wingate anaerobic test as a great example of the depletion idea, that again, when our phosphocreatine is providing energy, we can maintain relatively stable power outputs, but again, we talk about anywhere between 8 to 12 seconds our phosphocreatine system can last, and you can see that the change in slope typically occurs right in and around there, telling us that when we deplete creatine phosphate as a major source of energy, that fatigue will set in. We finished class on this slide, but I'll cover it again just to make sure. But this idea, looking at substrate depletion, primarily focused on carbohydrate metabolism. So we've talked about these three concepts here in the past, one of them being glycogen depletion during long duration endurance type events. I'll note you could deplete glycogen with really high intense exercise, but oftentimes we can't make people work out that hard. So most glycogen depletion scenarios happen over long duration exercise. Again, this is usually anywhere between 2 to 3 hours before we deplete our glycogen. Again, when we're talking glycogen, we are specifically talking muscle glycogen. So it's associated with fatigue, and indeed we can generally kind of see this, that in marathons we hear the suggestion that it's like the monkey jumping on their back, and that is usually associated with glycogen depletion. It usually is uniform depletion across different fiber types, so it doesn't matter. So that's kind of why the endurance exercise training, you're going to deplete glycogen in all of the fibers and cause fatigue. I briefly kind of mentioned we can deplete during really, really high intense exercise over a relatively short amount of time. It's hard to do. We can do it in athletes. When we do that, we can actually deplete it in specifically type 2 fibers, but oftentimes that's a challenge to be able to get done. So what I kind of take away is from that little dot is that when we're talking glycogen depletion with endurance training, we're talking about all fiber types fatiguing because of the lack of glycogen. We can try to impact that by carb loading beforehand, and then there's also other type of supplements or attempts to do this by trying to what we call spare glycogen. So one of the common ones is using caffeine to get more free fatty acids in to reduce carbohydrate utilization during exercise. If we have a depletion of blood glucose, again, in general, this is a very negative thing. Our body tries to maintain blood glucose in relatively very small window, both up and down. And during long duration exercise, we can fail to be able to do that, primarily because we aren't maybe taking in enough glucose, and we've depleted liver glycogen as well. So during short exercise bouts, blood glucose rises, so definitely doesn't cause a depletion effect during short high intense exercise bouts. But with prolonged activity, we can, and earlier in the semester we kind of showed the video of the guys at the end of marathons like staggering and stumbling and struggling to be able to even stand up. That's usually an indication that blood glucose has dropped below a normal value, and therefore they can no longer maintain homeostasis. Again, one of the primary reasons that these either a blood glucose drop or glycogen depletion causes peripheral fatigue is most likely associated with the decreased ability to produce ATP. And this is because of this concept of anaplerotic substrates. So what we've learned is the Krebs cycle is, again, you guys have all learned as classes that the Krebs cycle is a circle. So you start at the beginning, you produce a lot of intermediates in between, and ultimately you end up back where you started from. What we learned previously, to just reiterate, that there are parts of the cells that will take some of these intermediates and siphon them off in other reactions. So just reactions there. And what happens is, as we siphon off or kind of steal them out of the Krebs cycle, we start to lose the energy to produce this. The primary way that we can replace this is through pyruvate. Can fix this problem. So pyruvate can replenish these reactions, and of course pyruvate comes from glycolysis, which of course is the breakdown of carbs, so either glucose or glycogen. So if we run out of glucose or glycogen, we reduce our ability to produce pyruvate, which ultimately leads to a decrease in the Krebs cycle to produce ATP. And that's why you sometimes hear the phrase, fats burn in the flame of carbohydrates. We need carbohydrates in order to maintain this Krebs cycle. So if we jump over to the accumulation hypothesis, one of the primary and kind of best well-known looks at the accumulation hypothesis is the accumulation of hydrogen ions, which causes a change in pH. So here, if we look at blood pH, so blood pH, and we'll note, is a marker of a muscle pH, so they kind of mirror each other. But what we can see here is that blood pH is typically around 7.4. During exercise, and in this case these are high intensity exercises, we get a decrease in pH because we have an increase in hydrogen ion production, and we become more acidic. And you can see, again, sometimes this, even if you're in really high intensity exercises, this pH continues to persist. You never get super low on pH, so it's very rare to get below 7. I'll note you can maybe in skeletal muscle, but it's really unlikely. As we can kind of see here, but the kind of key takeaway from this figure is that pH decreases with exercise, and this can impact fatigue. So first, let's talk about what the source of hydrogen ions likely is. So many people, if you were to read them, would suggest that this is potentially from lactic acid. It's my professional opinion that lactic acid is not a major source of ions. Instead, the major source is the hydrolysis of ATP. So ATP, when we use it to produce energy during a muscle contraction, we use a hydrolysis reaction. And what this again uses is hydrogen hydrolysis via water, and in this process we break phosphate group off, are left with ADP plus PI, and in the process we have a hydrogen ion come off. Again, muscle contraction produces a massive amount of hydrogen ions because of the ATP demand, and therefore we drive acidity down here. I'm going to start at the bottom here because we're really focused on peripheral fatigue, but hydrogen ions have the ability to serve a primary role in peripheral fatigue. They may inhibit glycolysis through PFK, or phosphofructokinase, which is again is our rate-limiting enzyme. They may interfere with calcium binding to troponin C. So a change in pH actually impacts the protein structure of troponin C, such that calcium struggles to bind. And if you remember, this is basically one of the early steps of cross-bridge cycling. And therefore, we reduce force because of cross-bridge cycling. And it also may impact pain receptors, and I'm going to highlight this because for two reasons. One, it's thought that these pain receptors may be kind of what you think about when you exercise and you feel the burn, if you would. I don't know if this is scientifically proven that this is actually a change in pH, but there is the hypothesis and thought that we can stimulate pain receptors and you can feel the burn of the exercise here. Obviously, if we feel the burn here, what that ultimately relates to is our second point as I move up, which is a role in central fatigue. So central fatigue, again, is related to the central nervous system. Any type of discomfort through pain, potentially nausea or disorientation due to this change in acidity would result in reduced performance and central fatigue. There's actually a couple significant kind of biochemistry views of this in central fatigue as well. There's thought that it actually limits the ability of oxygen to bind with hemoglobin in the lungs, so we actually reduce the amount of oxygen carrying capacity with acidic blood. And it also has the ability to go outside of the muscle and impact hormone-sensitive lipase, which is why I have it kind of here, is because it's not actually impacting muscle, but it impacts hormone-sensitive lipase, which is in adipocytes, decreasing fatty acid oxidation, and ultimately this leads to increased carbohydrate metabolism, using more glucose, more glycogen, and leading to that depletion hypothesis. Again, hopefully you can kind of see that this depletion and accumulation hypothesis, both of them, it's hard to avoid those two, and because they play such an important role and work in many of the same mechanisms as we go through. The other part of the accumulation hypothesis relates to calcium. Calcium is impacted in at least 3 ways, and we'll highlight 3 of those here. The first is what we call a decrease in free calcium. Recall that calcium is stored in the sarcoplasmic reticulum. An action potential comes down the T-tubule, and we learned this in class. We learned that the way that we couple those, so we can couple the excitation part of the action potential to the contraction or mechanical part through a receptor known as DHP and RYN, dihydropyridine and ryanodine. So those two work together, and then that causes calcium to be released from the sarcoplasmic reticulum. So when calcium is released, it goes and binds to troponin C, moves tropomyosin out of the way, so that we can then start actinomyosin cross-bridge formation, power stroke, and muscle contractions. So when we look at that, again, as students up until this point, you probably just thought about that in a vacuum, that happens perfectly. We contract, and then when we relax, when the action potential goes away, all of that calcium is taken back up into the SR and ready to go again. But over prolonged exercise, we actually see that the amount of calcium in the sarcoplasmic reticulum will slowly decrease. And we call that a decrease in free calcium because the calcium isn't free to be part of muscle contractions and ultimately tells us it goes somewhere else. One of the primary spots we believe it goes is into the mitochondria. So the mitochondria are an organelle just like the sarcoplasmic reticulum, and during a long prolonged exercise, lots of calcium, some of it will go into the mitochondria and stay there. If there's more calcium in the mitochondria, that means there's less in the SR and therefore less to be used for muscle contractions. There's also suggestions that calcium coming out of the SR may be problematic with excitation contraction couplings, either at the T-tubules or sarcoplasmic reticulum. Again, what we're really talking about here is our DHP receptor, which is the part that is the EC coupler, and then interference with our ryanodine or RYN receptor. Again, these two basically work together to couple action potential and contraction. And so there may be a reduced ability to get calcium in because we have problems at the excitation part, or interference with the ryanodine receptor of allowing calcium to be released from the SR. The second is a decrease in calcium responsiveness. What we see here is, and you can see this here in this figure part 2, is that calcium responsiveness is decreased. So when we put all of our free calcium out, we actually see less contraction. So it has nothing to do with the calcium itself, it's just the muscle itself doesn't respond to that contraction with the same amount of force. We mentioned the primary reason for this, and the previous one is likely because hydrogen ions interfere with calcium binding to troponin C, which ultimately then reduces cross-bridge cycling and force production. And last but not least is number 3, which is a decrease in the sensitivity of calcium. What we can see here, and you can see it in this graph here, is that we shift the calcium curve to the right. And what this tells us is to get the same amount of force produced, we must put more calcium out. So more calcium must be present in order to get the same amount of contraction. To read that on that graph, you can kind of see here. So to get 50% of your muscle contraction, we need a calcium level of about 0.4 micromolars. To get 50 here during fatigue, we would need 0.6 micromoles. Again, need more calcium. This compounds with the idea that we have less free calcium available. Again, so we get a decrease. We get a decrease in the free calcium available. Means less force as we go through. So when we look at peripheral fatigue, we'll kind of summarize it here, and then I'm going to add 2 more into the accumulation. This one's got a little bit of extra words on it, but I kind of like the picture, and we can highlight some of the stuff we've already covered, and then add in one more part of that, and that's the reactive oxygen species side of things. So let's highlight what we've learned so that you guys can kind of follow along and not be hopefully too distracted by some of the excess stuff on here. Again, we talked about this right here, which is our voltage senses. So this is our DHP receptor here. So We can kind of see them here in the green, and we can ultimately see that one of the peripheral sensors, we have a decrease in voltage sensing part of this here. These here are what we would kind of consider our ryanodine receptors as well, which are going to help calcium be released. And you can see that here. So here's our ryanodine receptor. Again, we have a decrease in the amount of calcium released. We mentioned this as free calcium being available for several reasons as we kind of go through kind of looking at some of these. We can actually look at modification. So this is due to hydrogen ions impacting the function of that. So changes in pH cause it to not work correctly. We can also look at reuptake. So that's the release, the reuptake, and I want to highlight that here. So one of the things that I wanted to add for this is ROS. So we learned about this earlier in the class about reactive oxygen species can be important contributors to fatigue. They can decrease calcium, and I'm going to cheat and just say re-up. Again, we're highlighting that here. We can decrease calcium reuptake into the sarcoplasmic reticulum in order to then kind of complete this cycle of promoting calcium uptake and calcium release for contraction and relaxation. The second thing that we'll highlight is our myofibrillar proteins. Again, when we think these are actin and myosin. So reactive oxygen species decrease cross bridges. So they actually impact actin and myosin, reducing cross bridges and their ability to form. And then last but not least, we'll highlight on this slide that we talked about is that our calcium sensitivity goes down. So we reduce calcium sensitivity. So kind of adding again how calcium and ROS work together. And ultimately, when we kind of think about this kind of big long-term idea of fatigue, hopefully you can kind of gain the appreciation that this does make it very difficult to isolate those. It also gives us a lot of variables between individuals. And so when we talk about fatigue, we then relate it to individuals by talking about the fatigability. In other words, each person is individualized in their response to fatigue. We can't just go out and give one protocol and expect every person to fatigue exactly the same. So that inter-individuality in fatigue is defined as, is our definition of fatigability. And there's a handful of things that that are going to impact fatigability, primarily related to the individual and the environment. So things that we know can impact this is age. As you get older, you increase fatigability. So increased age leads to increased fatigability. Sex. Surprisingly, females are actually less fatigable than males, at least in long duration endurance events. So to get an increase in fatigability here, we say males have increased fatigability here. If you have neurological, again, this is going to relate to the central nervous system, but if you have some type of neurological health, that includes something like depression, decreased motivation, that will ultimately encourage you to be increased fatigable. So decreased neurological health increases fatigability. Hydration status is the last characteristic. If you are dehydrated, you increase fatigability. Environmentally, increased altitude leads to decreased fatigability. Or sorry, increased altitude increases fatigability. So the higher up, the faster you fatigue. The same exact with temperature and humidity. Again, oftentimes these are related to the central nervous system of I just don't want to do it when it's hot and humid outside as we live in Texas. So to finish up, here's our model of fatigue. So again, as we kind of look at fatigue, as we looked on this side, this is our central or our perceived fatigability, which is highly impacted by our psychological state, as we highlighted there, or homeostasis. So if we have some type of problem here, either with hydration, neurotransmitters would be kind of our neurological health, as we talked about, any non-blood glucose, ultimately those are going to impact our perceivability, fatigability, or our central nervous system. Our performance fatigability is, of course, our peripheral. Again, we primarily highlighted this side of the graph here of muscle activation through either the accumulation or the depletion hypothesis, kind of looking at both activation and contractile functions as we go through. Again, you can now see why fatigue is a mixed bag. It's a large amount of things that can ultimately lead to fatigue. It's really hard to study because many of them happen in the exact same time and same scenarios. But hopefully you've gained at least an idea of where the sites of fatigue are and can at least start to think about what types of exercise might cause those fatigue mechanisms to occur.